Hello, my name is Elena Stübi. I would like to present you a topic about neurotoxicity and neurotoxicity testing of medical devices. Medical devices possess risk to patients, and this risk has to be assessed. How is it done? It is usually performed within a risk management framework provided by ISO 10993-1 and ISO 14971. Since many different types of medical devices exist and there are different types of therapy delivery they provide, there is no specific sequence of biocompatibility testing and preclinical studies suitable to obtain regulatory approval. This means that the biocompatibility and safety assessments of medical devices, including neurotoxicity assessments, are done on a case-by-case basis. To identify neurotoxicants in medical devices is quite complex. Why is that? It's because a medical device is usually assessed in its final form, meaning packaged and sterilized. So it's not only the list of chemical constituents that are available in the medical device, but it's also packaging residuals, degradation products, cleaning and sterilization residuals that can play a role and bring neurotoxicants with them. One of the first questions that might appear during the biocompatibility evaluations is the human body exposed to neurotoxicants through a medical device, and if yes, what are they? This is a complex question to answer as well, because we are not dealing with pure chemicals, but we rather deal with chemicals and mixtures that may leach and migrate from a medical device into a human body at quite low concentrations. So what is neurotoxicity? Neurotoxicity is any modification to the structure and/or function of the nervous system, for example, brain, spinal cord, or body function controlling nerves, caused by the exposure to chemicals, biological or physical agents. It occurs following both acute or chronic exposures to either neuronal or glial cell types. Acute neurotoxicity occurs immediately after the exposure to the compound, or it can be delayed. Chronic neurotoxicity is usually due to repeated low-dose exposures. Which neurotoxicants can be expected from medical devices? On this slide, I'm showing you three possible groups of neurotoxicants: physical agents such as electrical current, chemicals such as ethyl hexyl phthalate or DEHP, and particles. In the next three slides, I will briefly explain you these three types of neurotoxicants. The electrical current. Electrical current can damage neurons and other tissues by several mechanisms: production of electrochemical reaction products, by induction of non-physiological levels of excitation, and by increasing the neuron excitation thresholds. Also, electrode heating can cause thermal damage. The thermal damage is shown on these four pictures. These are the results from a magnetic resonance imaging safety study of a swine spinal cord. Picture C and D shows the acute hemorrhage, inflammation and edema, indicated by arrows, red arrows, of the cord adjacent to the electrode. Pictures E and F show the formation of spheroids and axonal degeneration, indicated by black arrows. Another type of neurotoxicant is chemicals, and I would like to use as an example the substance which is called DEHP. So what is DEHP? DEHP is a phthalate used in medical devices as a plasticizer. Current scientific data on this substance indicates that it possesses anti-androgenic properties and is therefore considered to be an endocrine disruptor. Recent epidemiologic studies show that prenatal exposure to phthalates may be associated with altered genital and pubertal development in infants and children. Also, DHP may have an adverse impact on the development of the brain, and there is evidence that DHP can cross the placenta and enter the fetal circulation that might lead to the disruption of normal fetal brain development. Particles or particulate matter can be also considered as neurotoxicants in neurologic medical devices or generally in medical devices. It is important to assure that a medical device is free of submicron particles. What are particles actually? So particles or particulate matter are mobile particles other than gas bubbles present on or arising from the use of a medical device. Particles can arise from the manufacturing environment, the manufacturing process, the product itself, or from its packaging. Why is there a concern about them? This is because submicron particles cannot be readily detoxified or eliminated from the body, and this could lead to the biological responses such as inflammation. Because of the material properties such as geometric, physicochemical properties, or increased amount of chemicals that can be released from particles. Neurotoxicants can be expected from medical devices with a contact to central nervous system and peripheral nervous system. An example would be implantable spinal cord stimulator, and these types of devices are called Neurologic in this presentation. Medical devices, neurotoxicants can be also expected from medical devices without a contact to central nervous system and peripheral nervous system. An example would be blood transfusion bag or parenteral nutrition medical device. Neurologic medical devices are used to treat neurologic disorders or non-neurologic disorders. Examples of neurologic disorders are Parkinson's disease, migraine, stroke, pain, hydrocephalus, and examples of non-neurologic disorders are obesity, rheumatoid arthritis, sleep apnea, chronic heart failure, fibromyalgia. When a medical device manufacturer aims to develop a neurologic medical device, it is desired to have a material that is soft, non-toxic, non-carcinogenic, non-antigenic, non-mutagenic, non-pyrogenic, pro-healing, and electrically conductive. The problem with this list is that currently ideal material does not exist. On this slide, I put a list of materials, coatings used in neurologic medical devices, just an overview. You can read and then understand how complex medical devices are. Let's have a look on some neurologic medical devices. On this slide, a spinal cord stimulator, which is a class 3 medical device, is shown. Spinal cord stimulators produce electrical pulses to relieve neuropathic pain in patients for whom conservative treatment has failed. Another example of a neurologic medical device is a cerebral shunt. This device is used to treat hydrocephalus, and hydrocephalus is the swelling of the brain due to excess buildup of cerebrospinal fluid. So far I spoke about the neurologic medical devices, the devices which are in contact with CNS and PNS. On this slide I am showing you medical devices without contact to CNS and PNS that may release neurotoxicants. They are blood transfusion bags, parenteral nutrition medical devices for adults and for neonates. They are classified as Class 2B medical devices in Europe and Switzerland. One of the neurotoxicants that may be present in the mentioned devices is phthalate, DEHP. According to the FDA, there were some recalls of such products in the past due to the presence of this substance, and what is more important, this substance was not labeled on a medical device. There was and there is a lot of noise due to phthalates currently. In 2016, Scientific Committee on Emerging and Newly Identified Health Risks published an opinion on the safety of medical devices containing DEHP. This report or opinion reported that procedures such as exchange transfusion of blood in neonates, total parenteral nutrition in neonates, and hemodialysis may lead to high exposure to DEHP, potentially leaching from the medical devices used. Also, according to the opinion, the tolerable daily intake TDI for DEHP is set as 50 microgram per kilogram body weight per day based on the EFSA calculations. Important to know about the DEHP is that the bioavailability of this substance following parenteral exposure is considered to be 100%. According to the International Agency for Research on Cancer, this substance is classified as Group 2B carcinogen, meaning possibly carcinogenic to humans. Now, exposure to DEHP may significantly exceed the TDI in two types of patients, and this is based on the opinion published in 2016. Adults undergoing hemodialysis with a 40-fold higher exposure than the TDI, as well as neonates in neonates intensive care units with 120-fold higher exposure than the TDI. It is really an unacceptable situation, and something has or had to be done with it. On this slide, I'm showing a brief overview how biocompatibility testing is performed in medical devices or neurologic medical devices in Europe and Switzerland. This testing is directed by ISO 10993 series. It always starts with chemical characterization of the finished product, guided by ISO 10993-18. Then, usually toxicological risk assessment of medical devices components is performed, and this work is guided by ISO 10993-17. By the way, the updated standard is going to be published soon by ISO. After these two steps, an assessor is able to have a defined further biocompatibility strategy. And this strategy usually includes tests from ISO 10993-6, which describes tests for local effects after implantation, and especially one annex is important in terms of nervous system. It's Annex D, that describes tests for brain tissues. Also, ISO 10993-4 is used in this strategy, and it provides selection of tests for interactions with blood. After defining the biocompatibility strategy for some devices, and especially for Class 3 medical devices, preclinical safety testing strategy has to be defined and performed. For neurotoxicity testing, OECD guidelines may be very helpful. I give you here the list of existing OECD guidelines. I'm not going to talk about them, but maybe if you're interested, you can go through them or contact the publication I have taken this list from for further information. Biocompatibility and safety risks of medical devices and neurologic medical devices should be identified and may include hypersensitivity to materials, chemical toxicity, mechanical damage, potential changes in material composition due to manufacturing processes, and in vivo degradation or electrochemical reactions, for example, metal dissolution secondary to charge-driven processes. What is so peculiar about the testing strategy for neurologic medical devices? It is potential delamination or corrosion of coatings can introduce foreign material into tissues. It's important to know. The pathologist assessing biocompatibility must consider that the entire pathway of implantation may contain exfoliated device materials with potential inflammatory reactions. And the last but not least, for the devices with subdural implantation sites, cerebrospinal fluid redistribution can lead to foreign material deposition in locations distant from the pathway of implantation. Usually, the neurotoxicity testing as part of regulatory requirements for medical devices, and neurologic medical devices especially, starts with in vitro testing. This testing provides information, but this information is not enough. Still, key models for understanding tissue-device interactions are in vivo models. Both in vitro and in vivo models have to be done under GLP conditions. What is really desired nowadays are the alternative testing methods. But we are not there yet. What I mean by alternative methods, it is actually a combination of in vitro and in silico methods. For in vitro methods, it is important to have 2D and 3D cell systems that can show the complexity of the nervous tissue and that can show the response to toxicants. These cell systems should be also able to present the interactions between neurons and glial cells. In silico methods are usually QSAR and toxicokinetic models that predict ADME and blood-brain barrier penetrations. As I said, we are not there yet at the moment, but hopefully we will be there soon. For such testing, it's also important to have a histopathologist involved in order to understand the tissue responses. With this, I am at the end of my presentation and would like to thank you for your attention. The RMS Foundation would be happy to discuss and work with you on projects related to toxicological risk assessments of medical devices. Please feel free to contact us.